Hi, everybody, and welcome to this week's lecture. What we're going to try to accomplish today is we're going to go ahead and take a look at the structures and functions of some of the major eukaryotic and animal cell components. Now, when it comes to a eukaryotic cell, remember EU meaning true, so EU equals true, meaning that there is going to be a true nucleus. And you'll notice here on this diagram that we have many different components when it comes to all of our organelles. Remember, organelles just essentially means all of the different pieces that are within our cells right here that are going to help with regulating cell functions. So some of the major ones we're going to identify and look at are going to be the cytoplasm. We're going to look at the cell membranes, the nucleolus, nuclear envelope, nucleus, nuclear pores, both the smooth and rough endoplasmic reticulums. We're going to also look at the Golgi apparatus. We're going to look at some lysosomes, and then we're also going to look at the mitochondria and ribosomes. So one of the first things that we need to understand and we need to look at first is exactly what's going on in the inside of these cells and how is everything going to be suspended. And that's where our first organelle is going to come into play, which is going to be known as the cytoplasm. Now, the cytoplasm is just going to be the area between all of our organelles and membranes. So our cytoplasm is going to suspend all of our organelles, and it's going to be in this jelly-like substance, which again is going to be about 70 to 80% water, but it is going to be semi-solid as well due to its consistency because of proteins that are going to be contained within it. Again, the cytoplasm's main job and main idea is to kind of just suspend everything and keep everything where it needs to be. Our next organelle is going to be the plasma membrane, or in this case, the cell membrane. And one of the important things we need to remember about the plasma membrane is that it has that phospholipid bilayer. Now, remember, this is going to be selectively permeable, and that's going to be a very important characteristic when it comes to this phospholipid bilayer. Remember, the selectively permeable membrane allows certain things to come in right here and certain things to leave. So think about this entire bilayer like a gate. And again, this is going to be kind of like a security guard dictating what's going to come in and what's going to leave. Some things are not going to be able to go, but again, some things are going to be able to pass through. Now, when it comes to this phospholipid bilayer, remember the reason why it's known as a bilayer is because one of the layers is made with the hydrophobic and hydrophilic ends of our lipids, and the other part is going to be another piece as well. So remember, bi, just like a bicycle, contains one, two pieces. And this can be found in many other places as well. You can see these in our nuclear envelopes and our endoplasmic reticulums. So again, our phospholipid bilayer contains two pieces. You can see this is our first piece right here. And you can see that this is our second piece over here. And again, its main job is to act as a barrier between the inside and outside of the cell. Our next organelle is going to be the nucleus. Now, the nucleus is going to be very important because it's going to contain all of our genetic material, also known as our chromosomes. So again, right here in the very middle is going to be where our DNA is going to be stored. Now, again, this is really important, and the reason why this is known as the control center is because this is where all of our genetic information is that contains and codes everything for our body. So things are like our eye colors, hair colors, protein structures are all going to be found inside of our nucleus. Now, usually there's only going to be one per cell, and these are going to be bigger, just this itself, larger than our prokaryotic cells. Remember, prokaryotic, pro equals no, meaning that they don't have a true nucleus. Now, we are going to have a couple pieces when it comes to our nucleus. We are going to have our envelope, so this right here. And essentially, all that's going to be is a double membrane that's going to separate the DNA from the cytoplasm. Remember, DNA is going to be our genetic information, everything that's going to code for the body. And then the cytoplasm is going to be that semi-solid liquid that is going to suspend all of our organelles. Now, we're also going to have something known as our nuclear pores. And essentially, all that's going to be are little areas that are going to allow things to move back and forth. When it comes to that, as we are going to continue and learn more about DNA in future lectures, these pores are going to be important for allowing RNA to leave. And the reason why is because when we get to protein synthesis, when we get to things like that, transcription translation, DNA is going to be much too large to leave because, again, we want to keep our DNA inside of our nucleus. But when we're trying to produce these proteins, we need to send a message out. So in this case, we're going to produce something known as mRNA, and it's going to allow or be able to leave the nucleus because it's going to be small enough to leave those pores. So again, the nucleus, a couple important things. The nucleus inside is going to contain our DNA. Envelope outside is going to, again, separate the DNA from the cytoplasm. And then our nuclear pores are going to allow things to come in and out, very, very specific things to come in and out. Oh, and then the nucleolus is going to be a region inside of the nucleus where the ribosomes are going to be assembled. Now, speaking of ribosomes, these are going to play a very important part as well. And ribosomes are going to be located inside of the cells, and they're going to be made out of two different subunits. They are going to be made of special RNA, specifically known as our rRNA, ribosome RNA, and proteins. Now, looking at this over here, you can see that this right here, all the way around here, is going to be known as our ribosome large subunit. And then down here, this is going to be our small subunit. So you can see it's made out of those two different pieces. And one of the major jobs that the ribosome is going to do is it's going to allow for protein synthesis. All that basically means is our mRNA, remember, which is going to leave the nucleus. So our DNA is going to make our mRNA. It's going to leave the nucleus, and it's going to come to our rRNA. And then the mRNA is going to run through or in between both of those two different subunits. And then what's going to happen is our tRNA, known as our transfer RNA, is going to come down here, read this spot, and it's going to start assembling our amino acids. Once it's assembled our large chain, it's going to produce our polypeptide. So again, ribosomes, the main thing for this is to allow for protein synthesis. And again, this is just a big word to kind of just essentially denote the production of proteins. The next organelle we're going to look at is going to be known as our mitochondria. Now, when it comes to the mitochondria, I'm sure you've already heard this, that it is going to be the powerhouse of the cell. Now, I'm sure you're probably wondering, well, why is it going to be known as the powerhouse of the cell? And the reason why is because it's going to go through something or a process known as cellular respiration. What it's going to do here is it's going to go ahead and take energy. So it's going to take carbohydrates, it's going to take oxygen, and it's going to go ahead and produce ATP, something known as adenosine triphosphate. Again, it's going to go ahead and go through the cellular respiration process. It's going to utilize energy, and it's going to produce ATP. Now, the way that it's going to do that is the mitochondria itself is going to have many different structures that are going to allow for it. And we're not going to get too much into this. We will in future lectures. As of right now, we're just going to identify certain players or certain structures that are going to allow for the ATP production. And the major one we need to look at or remember is going to be the inner membrane folds. Now, these folds are going to be known as cristae, and you'll notice that the inner folds right here are going to be all of these pieces over here. Okay, so looking all the way around over here, that is going to be our cristae. And then we also have something known as our mitochondrial matrix, and it's going to be this blue substance all the way around here. So again, our mitochondria is going to be very important because it's going to allow for ATP production due to cellular respiration. And one of the cool things is if you can think back to our last lecture, the mitochondria also has its own separate piece of DNA. So this kind of supports the idea that this was a prokaryote that was taken in by our eukaryotic cells. So we have two sets of DNA, and this again is allowing for the ATP production, which allows us to have that energy to utilize and do whatever we need to do. All right, so this is going to be our first test for knowledge. There are going to be a couple parts in this, so I'm not going to kind of go over everything. I'm going to give everybody about a couple seconds to pause this and go ahead and try this out, and then we'll go ahead, check out the answers, and look at it together as a class. All right, welcome back. So I'm only going to go ahead and kind of do two of these. So the first one we're going to go ahead and look at is going to be this right here. So which of the following cell structures is most active during vigorous exercise? So in this case, out of all the organelles we've currently talked about, one of the major ones is going to be the mitochondria. And remember, the reason why is because it's going to go through cellular respiration in order to produce ATP. And remember, ATP, adenosine triphosphate, is going to be a very energy-rich molecule, which is going to allow us to do whatever we need to do. In this case, whenever we're utilizing vigorous exercise, we're going to be creating or going through cellular respiration in order to create our ATP. The other one I do kind of want to go over is this right here, based on your knowledge from prior lectures. What do you think the membrane surrounds the cell, the nucleus, and mitochondria are primarily composed of? Again, it's going to be our phospholipids. Remember, we have our hydrophobic and hydrophilic ends in order to create our lipid bilayer in order to allow for that separation of the outside and the inside. All right, so let's go ahead and continue. The next thing we're going to look at is going to be our endomembrane systems. Now, essentially, our endomembrane systems are going to contain our internal membranes, just like the name suggests. And what's going to happen is they're all going to work together to do a couple things like modify, package, and transport lipids and proteins. Again, all of these systems, these endomembrane systems, are going to work together to modify, package, and transport lipids and proteins. Now, this is going to include all of these membranes, our nuclear envelope, our lysosome, vesicles, endoplasmic reticulum, which we are going to have two of these, specifically our smooth endoplasmic reticulum and then the rough endoplasmic reticulums. Both are going to do very specific and important things, the Golgi apparatus and our plasma membranes. So let's go ahead and dive into our endoplasmic reticulums. So let's go ahead and see exactly what they're going to do. As you know, there are going to be two different types. We're going to have our rough ER, and just like the name suggests, looking at our ERs, which are going to be all of these right here. The rough ER are going to have all of these little circles on them. And all of these little circles are going to be our ribosomes. So remember, ribosomes are going to be responsible for protein synthesis, or the actual production of our proteins. Now, when it comes to the smooth ER, these are going to be responsible for our lipids. And again, just like the name suggests, we are not going to have any of those circles around it. So it's going to be very, very smooth. So again, the rough ER is going to look bumpy. It's going to have all of our ribosomes. And then our smooth ER are not going to have anything on them themselves. Now, one important thing is the ER tubules, which are going to be all of these pieces here, here, and here, are going to be known as our lumens. And again, the membranes of the ER are going to be continuous with the nuclear envelope. So again, you can see the nucleus here, nuclear envelope, and it's going to be continuous from that because it's going to essentially be an extension from it. So let's go ahead and dive into our rough endoplasmic reticulum, or in this case, the rough ER. Now, again, the main idea from this is to produce proteins. We have a bunch of ribosomes attached to these. So again, they're going to be responsible for the manufacturing of these proteins. So new proteins fold in the lumen of the rough endoplasmic reticulum. And then proteins are either going to be incorporated into the cell membranes or secreted from the cell. In this case, protein hormones and enzymes. So proteins are not destined to stay in the rough ER. Their destination can be transported via transport vesicles that bud from the ER membrane. So again, it's going to help transport some of those pieces. The next type is going to be the smooth endoplasmic reticulum. And remember, the main difference between them is the rough ER has all of our ribosomes on them, and our smooth are not going to have any of those pieces on there. So the smooth endoplasmic reticulum is continuous from the rough endoplasmic reticulum, and it's going to have few or no ribosomes. So again, remember, the easiest way to kind of see this is the rough has those ribosomes, so it's going to physically look rough, or it's going to have all those dots, versus the smooth is not going to. Now, the smooth is going to be responsible for the synthesis, or in this case, the production or make of lipids, including steroid hormones. It's also going to be important for detoxification of medications and poisons, and it's going to also store calcium ions. Our next organelle is going to be the Golgi apparatus. So when it comes to the Golgi apparatus, it's going to be pretty important because lipids or proteins within the transport vesicles still need to be sorted. So in this case, the Golgi apparatus is going to go ahead and do that. It's going to sort, package, and tag all of them. So this is all happening right here. And if you go ahead and look at this, you can see in this graphic, we have our proteins that are going to be exported. So remember, the ribosomes are producing them. So they export it out over here into our transfer vesicles, which are going to move into our Golgi apparatus. And again, it's going to enter our cis phase right here. And then it's going to exit our trans phase, which is the back or opposite end. So think about the cis, which is going to be the front. Let's see, fronts. And then our trans, which is going to be the back. And again, the entire idea from this is that it's going ahead and it's packaging this protein and it's helping it transport it out to wherever it needs to go. So again, transport vesicles from the ER fuse with the cis phase and empty their contents into the lumen of the Golgi. Proteins and lipids travel through the Golgi on their way to their final destinations. So again, think about the Golgi apparatus as, let's say, the post office. It's going to be packaging, tagging, sorting, doing all those things to make sure that it's getting to exactly where it needs to go. The next thing we're going to kind of talk about is going to be known as our extracellular surfaces, or in this case, our extracellular matrix. And the reason why we're going to kind of look at this is we focus a lot on the inside, and this is kind of a preview of what we're going to see in future lectures. But in this case, cells in the most multicellular organisms are going to release materials into this extracellular place. So primary components of these materials are going to be proteins, specifically going to be proteins in collagen. So it's going to allow us to kind of like bind together. So here are collagen fibers. We can see what that's going to look like. It's going to contain our polysaccharides, proteins, and carbohydrates. And again, this is all going to be located outside, extracellular, again, outside of the cell. So extracellular matrices hold the cell together to form a tissue. So remember, we're going from cells to tissues, and eventually we're going to move further and further up our chain. So again, you can see our extracellular matrix are going to hold all of our cells together in order to produce our tissues. All right, so this is going to be our second test your knowledge. So what I'm going to do is I'm going to go ahead and give everybody a couple seconds to pause this, and then we'll go ahead and check out one of these and then move on to our last slide, and that will be it for our lecture. All right, so welcome back. And what we're going to do here is we're going to go ahead and take a look at number one. Which of the following is active in liver cells that are detoxifying? Alcohol detoxifying, remember, is going to be an important job. Golgi apparatus, the only thing it's doing here, just like a mailroom, it's just going to be sorting, packaging, sending out, so it's not going to be this. Rough ER, remember, is going to be the production of proteins and things like that. So it's not going to necessarily be that. Mitochondria is going to be ATP. It is not going to be that. Nucleus has our DNA, so it's not going to be that. So in this case, it is going to be our smooth endoplasmic reticulum, which is going to be responsible for, remember, detoxifying things like medications, poisons, things like that. In this case, it is going to be detoxifying alcohol. All right, so the last thing that we need to kind of talk about is cell-to-cell communication. So one thing that's really awesome is cells do kind of communicate with each other. They like to know exactly what's going on, what states they're in, what's happening, if one of them's going through cell division, things like that. So within multicellular organisms, cells do communicate, and they're going to do two ways. The first is going to be a direct contact via a junction, gap junction. And then our second can be cell-to-cell recognition via interacting molecules of the surface. So in this case, for our first one where it says direct contact, just like the name suggests, they're going to be in direct contact with each other. In this case, this is going to be a gap junction within animal cells. You can see they're directly touching, so they're able to communicate and send things to each other. Same thing with plant cells. They're going to be in direct contact with each other. So you can see they're going in, going out. So they're communicating, so they know exactly what's going on. This one over here knows exactly what's going on with number 2 over here. Same thing with this. Cell 1 and cell 2 are going to be in constant communication, knowing exactly what's going on, what state they're in, and things like that. Cell-to-cell recognition is going to be this over here. You can see that we have these proteins on top of the cells, and we do have these receptors. So whenever they go ahead and come in contact with each other, this is when they're going to be sharing that information. So again, you can see that they're going to come together. And remember, these are always going to be very specific to each one, just like a lock and a key. And once they go ahead and get together, they'll be able to communicate and share that information, knowing exactly what's going on with each other. All right, and just like that, that is going to conclude this lecture for this week. So again, if you do have any questions, comments, concerns, please feel free to reach out to me, and I'd be more than happy to answer whatever questions you have. I look forward to seeing everybody in the next lecture.